Greetings and welcome to Cassava Science Clinical Conference Call. During the presentation, all participants will be in a listen-only mode. Afterwards, we will conduct a question-and-answer session. At that time, if you have a question, please press the 1 followed by the 4 on your telephone. If at any time during the conference you need to reach an operator, please press star 0. As a reminder, this conference is being recorded today, Monday, September 14, 2020. Your speakers today are Remy Barbier, Cassava's President and Chief Executive Officer, Dr. Burns, Senior Vice President, Neuroscience, and Dr. Friedman, Chief Medical Officer. Also joining us today is guest speaker Dr. Gonzalez Rojas. Dr. Gonzalez Rojas is a principal investigator of the company's Phase 2B study with Sumifilam in Alzheimer's disease. I would now like to turn the conference over to Eric Schoen, Cassava's Chief Financial Officer, for introductory remarks. Please go ahead, sir.
Thank you, France. Good morning. Thank you all for joining today's call. First off, I would like to note that today's presentation is available in the Investors section of the company's website at www.cassavasciences.com. I'd also like to remind you that any statements made during this call, other than statements of historical fact, are considered forward-looking statements and, as such, are subject to risks and uncertainties such that actual results could differ materially and adversely from those anticipated or implied in the forward-looking statements. Those forward-looking statements include, without limitation, statements regarding prior or current results of our clinical studies and plans for future clinical studies, as well as the various risks described in the company's annual report on Form 10-K for the year ended December 31st, 2019. Accordingly, you should never rely upon forward-looking statements as predictions of future events. Importantly, this conference call contains time-sensitive information that is accurate only as of the date of the live call, September 14th, 2020. Except as required by law, Cassava undertakes no obligation to revise or update any forward-looking statements to reflect events or circumstances after the date of this call. Before we begin, let me review the format for today's call. Remy Barbier, President and Chief Executive Officer, will open the call and presentation. Afterwards, we will open up the call for a question-and-answer session. Now, let me turn the call over to Mr. Barbier.
Thank you, Eric. This morning, we're super excited to present final results of a Phase 2B study of Sumifilam in patients with Alzheimer's disease. As I hope you know by now, in a clinical study funded by the NIH, Sumifilam significantly improved an entire panel of validated biomarkers of disease in patients with Alzheimer's. Today is a very good day for Alzheimer's patients. The ability to improve multiple biomarkers from distinct biological pathways with a single drug, to the best of our knowledge, that has never, ever been shown in patients before. For this reason, we are very excited to present the clinical results of Phase 2B. In order to facilitate the presentation, we will be following the slideshow, which can be found on our website. I am now on page 3 of the company presentation. Slide 3 shows a great lineup of presenters, including Dr. Friedman, Dr. Burns, Eric Schoen, and a guest presenter, Dr. Gonzalez Rojas, who is a principal investigator of the Phase 2B study down in Miami, Florida. I'm now on slide 4. Our mission is very singular and very dedicated. We are entirely dedicated to the development of novel approaches to both detect and to treat Alzheimer's disease. I'm now on slide 5. Our initial focus, for those who know us, is on developing Sumifilam, formerly known as PTI-125. Sumifilam is the first of a new class of drugs that target filament proteins. Filament-binding proteins are brand new to Alzheimer's research and we think can represent a very important advance towards the goal of ultimately disease-modifying drugs. Last year, around this time, we showed that Sumifilam reduced neurodegeneration and neuroinflammation in Alzheimer's disease. The results were published, both CTAD and a peer-reviewed journal, but the critique at the time was that it was an open-label study, so perhaps needed to be replicated. We absolutely agreed, and in fact, this year we completed a randomized, placebo-controlled phase 2b study of Sumifilam in Alzheimer's patients, thanks to an NIH grant. And the point of today's presentation is to inform our stakeholders of the results of this study. Ultimately, of course, our goal is to conduct a phase 3 study with Alzheimer's disease with Sumifilam. I'm now on slide 6. I think by now we all know that Alzheimer's is a progressive brain disorder, pretty much destroys everything in its track, including memory and thinking skill and many other things. It's a huge disease. Probably 5 to 6 million people live with Alzheimer's today, and that incidence, as far as we can tell, is expected to double. The take-home message is right now, today, there are no disease-modifying treatments for patients with Alzheimer's disease. I'm now on slide 7. Now, before we can actually dive into the final results, we have to step back a little bit. As previously reported back in May 2020, an outside lab did an initial bioanalysis of the phase 2b data. That data made no sense whatsoever. At the time, we put out a press release pretty much saying it made no sense. But what we've had time to do since that press release is what's called a post hoc analysis. And what the data shows is that just in the placebo patients alone, patients who only took placebo over a month, you would expect to see very small changes in biomarkers over a month. Instead, the initial lab showed data with huge dramatic swings going in both directions over a month. That is unheard of. In addition, the biomarkers generally move in the same direction because Alzheimer's itself is a disease of biomarkers. However, the initial bioanalysis showed biomarkers all over the map. It almost like it looked like the data followed a drunken random walk. Some patients even moved in opposite direction from each other. Some biomarkers moved in opposite direction. Generally, because Alzheimer's is a disease of biomarkers, these biomarkers move in tandem. They move together, almost in lockstep. And so one would expect to see robust statistical correlations. We saw none of the above. In fact, what we saw is that the movement in the biomarkers over 28 days were absolutely uncorrelated. For those of you with a statistical mind, the R2 value was 0.06, which essentially says a random walk. Again, in the spirit of transparency and full information, we included more statistical data on the placebo patients in the appendix. If you're really interested, I urge you to consult that. We're now on slide 8. An academic lab conducted a second and final bioanalysis of the phase 2 data. The academic lab showed what we considered to be valid, proper, and expected data in placebo patients. Data from this lab shows modest swings, some swings, of course, maybe 4 to 6% on average, in changes in levels of biomarkers over 28 days. But more importantly, they showed biomarkers that generally moved in the same direction and robust statistical correlations among changes in levels of biomarkers. For these reasons, both ourselves and our advisors and pretty much anyone we've shown all the data to have confirmed that the second bioanalysis is a valid analysis. With that short intro, I turn it over to Dr. Burns for a discussion around the science of Sumifilam.
Good morning, everyone. I'm excited to take you through the mechanism of action of Sumifilam, how it works, what it does. Sumifilam is our proprietary small molecule drug candidate to treat Alzheimer's disease, and this program has benefited from long-term support of the National Institute on Aging of the NIH. Sumifilam has a dual mechanism of action. It reduces both neurodegeneration and neuroinflammation, but it does these two things by binding a single protein target. And this year we concluded a comprehensive phase 2 testing program, which was conducted in consultation with our advisors and funded by NIH. Moving to slide 11, the target of Sumifilam is an altered conformation of the scaffolding protein filament A. You may have heard of filament A for its cross-linking with actin, as it's pictured here, to give the cell structure and motility, but it actually interacts now with over 90 different proteins and therefore influences many different signaling pathways. This is a large protein. It protrudes inside the cell. It's bound in the cell membrane, but it hangs down inside, interacting with a lot of different proteins. Now, in Alzheimer's disease, this filament A protein exists in an altered shape, and this altered shape is critical to the toxicity of beta amyloid. Moving to slide 12, to understand the mechanism of action of Sumifilam, we first have to start with understanding the filament A proteopathy, sometimes called a proteinopathy. A proteopathy is any distorted, dysfunctional protein that leads to disease. So altered filament A really acts as an accomplice to the toxicity of A-beta-42, the most toxic fragment of beta amyloid. And it allows A-beta-42 to signal, to basically hijack two receptors and signal through them to wreak havoc in the brain. The first is the alpha-7 nicotinic acetylcholine receptor. This is a receptor that's usually used for memory. Altered filament A scoots up to this receptor from the inside of the cell to allow A-beta-42 to bind it incredibly tightly. It binds with femtomolar affinity. And this binding allows A-beta-42 to steal the signaling machinery of this receptor and hyperphosphorylate the protein tau. That means it puts too many phosphate groups on the tau protein. Well, that's a problem because tau has an important job in neurons. It stabilizes microtubules, which are the conveyor belts of neurons, moving things around. And when tau can't do its normal job, things fall apart and the neuron dies. That's the neurodegeneration part. In a similar fashion, altered filament A links up to the innate immune receptor TLR4 to allow A-beta-42 to basically flip the on switch for this receptor and persistently release inflammatory cytokines, causing sustained neuroinflammation. So Sumifilam comes along, preferentially binds this altered form of filament A and puts it into its normal shape. And now in its normal shape, it breaks up from these receptor linkages. A-beta-42 releases the tight grip on these receptors. The signaling stops. So through a single target, Sumifilam reduces both the neurodegeneration caused by hyperphosphorylated tau and the neuroinflammation of the activation of TLR4. And slide 13 is a depiction of the over 10-year development of this program, starting in around 2008 with some basic science around filament A. Then we moved to an iterative screening, in vitro and in silico screening process, med-chem, lead profiling, and finally to in human trials in 2017. All this science, now into slide 14, has been published in peer-reviewed journals, starting in 2012, showing that filament A was critical to this toxic signaling, 2017 showing that filament A was in an altered conformation, and this year with the publication of our phase 2A clinical results. Sumifilam has also stood up to many peer-reviewed NIH grant applications and has received multiple NIH grant awards. So with all this science holding it up, moving to slide 15, we were able to come up with the clinical hypothesis: can Sumifilam provide early clinical evidence of disease-modifying effects in a well-controlled study? So our study objective, entering the phase 2B trial, was to evaluate safety, of course, and biomarkers, which are evidence of disease modification, as well as cognition following a 28-day treatment of this new first-in-class therapy. Moving to slide 16, so we relied on biomarkers. What are biomarkers? A biomarker refers to any objective, quantifiable measure of disease or disease severity. This is hard evidence for the presence of the disease and how bad it is. Typically, drug development has followed a one drug, one biomarker paradigm, and we are disrupting that paradigm because we believe that our drug can improve an entire panel of biomarkers indicating the neurodegeneration, neuroinflammation, and the signature of Alzheimer's pathology in these biomarkers. So I will now hand it over to Dr. Friedman to introduce you to the Phase 2b clinical trial.
Good morning. Before I start with the design of the Phase 2b study, I'd like to give you a little bit of a background, how we arrived at that study, why did we design it this way, and how did it go. So we conducted 3 clinical studies with Sumifilam. The first study was in normal human volunteers, where we wanted to look primarily at the safety of the product, but also how it is being distributed in the blood, the pharmacokinetics, how long it lasts in the blood, and we also used higher doses that we use in patients to make sure that it's safe to go into patients. The second study, we elected to do an open study in patients, in Alzheimer patients, basically to look at similar parameters that we look in the volunteers. We wanted to see how it distributed, how long it lasts, what would be the levels, and to give us an idea how to really dose in our more definitive studies as we keep going. So now we can go to the Phase 2b study. In that study, we elected to have patients that they have an MMSE or the mini mental state exam between 16 and 26. These are mild to moderate Alzheimer patients. We used 64 patients in 9 different U.S. sites. The key inclusion criteria in that study was looking at the tau to A beta ratio in the CSF or the cerebral spinal fluid, and it had to be greater than 0.28. The design of the study was double-blind, randomized, placebo-controlled, and had 2 doses of Sumifilam, 50 milligram and 100 milligrams. There were 2 major endpoints in the study. The primary endpoints was look at the panel of biomarkers and how they react after 28 days of treatment with Sumifilam. And the second endpoint was to look at cognition. I will now turn the presentation to Dr. Burns.
Did you want to cover the safety?
Oh, yeah. I'd just like to mention that Sumifilam was well tolerated in this study, as it was in all studies. There were no serious adverse effects. No patient dropped because of side effects from the study, and there were more side effects on the placebo group that were in the drug groups. Now to Dr. Burns.
Okay, I'm picking back up on slide 20, showing the first 2 biomarkers. These are total tau and p-tau 181. Now, I've already told you that tau is an intracellular protein, so when you find it in the CSF, that means the neurons have died and spilled it into the surrounding fluid. And this is a marker of Alzheimer's disease, but also of neurodegeneration. Now, p-tau 181 is a specific Alzheimer's disease signature, also spilled into the CSF when cells die, but the level of phosphorylation at that site on tau tells you it's Alzheimer's disease. And with these 2 core Alzheimer's disease biomarkers, both the 50 milligram dose group and the 100 milligram dose group significantly reduced total tau and p-tau 181 compared to placebo. The reduction in total tau was 15% and 18%, and the reduction in phospho-tau, p-tau 181, was 8% and 11%. Moving to slide 21. The third core Alzheimer's disease biomarker is A-beta 42. Now, it's actually low in Alzheimer's disease patients, and this is one way people diagnose Alzheimer's patients. So you want it to increase, and it also, it's slow because it's stuck to this alpha-7 receptor and other tight binding sites, and Sumifilam releases the A-beta 42 into the surrounding fluid so it cannot perform the toxic signaling. You see here that the 50 milligram dose group increased CSF levels of A-beta 42 by 17%, and the 100 milligram dose group increased A-beta 42 in CSF by 14%. These are both significantly different from placebo, which was unchanged. On slide 22, another method of diagnosing Alzheimer's disease is this total tau to A-beta-42 ratio that Dr. Friedman mentioned. And you can see that both dose groups of Sumifilam significantly decreased that diagnostic ratio, whereas in the placebo group, there was no change whatsoever. On slide 23, sometimes people use the ratio of p-tau to A-beta-42 to diagnose, and this ratio, too, was significantly decreased compared to placebo by 20 and 21%. Now on slide 24, let's look at neurodegeneration. In addition to the total tau indicating dying cells, we have two proteins, neurogranin and neurofilament light chain. These are both intracellular proteins. Neurogranin is found in the dendrites, neurofilament light chain in the long axons. And both the 50 milligram and 100 milligram dose groups significantly dropped both of these markers of neurodegeneration after 28 days of treatment. Neurogranin was reduced by 36% in the 50 milligram group and 43% in the 100 milligram dose group. Neurofilament light was decreased 28% and 34%, and of course, significantly different from placebo. Slide 25 shows you biomarkers of neuroinflammation. We looked at 3 different neuroinflammatory biomarkers. The first is called YKL-40. This is a marker, it's a glycoprotein that's increased during neuroinflammation, and it's involved in tissue remodeling after injury, and this marker was reduced by 10 and 12% in the 2 dose groups. Similarly, IL-6, interleukin-6, an inflammatory cytokine, was also reduced by both dose groups by 10 and 11%. Now, soluble TREM2 is a more recent biomarker that is getting a lot of attention recently. This is a receptor on microglia that are massively activated and responsible for neuroinflammation. And when these microglia are activated, this receptor is shed into the soluble form of TREM2, which we are measuring in the CSF, and it indicates active microglia and active neuroinflammation. This soluble TREM2 marker was decreased by 43% and 46% in the CSF of these Alzheimer patients taking Sumifilam. So together, this shows strong evidence of suppression of neuroinflammation. Now, on slide 25 is a visual of all the biomarkers we are presenting today, the increase in A-beta-42 and the reductions of the total tau, p-tau, the neurodegeneration markers, and then inflammatory markers. So slide 26, I'm sorry. So now let's move to slide 27. We also measured cognition, even though this was a very small trial, and we measured cognition because our preclinical data in transgenic mice and Alzheimer's postmortem brain tissue shows that we improve receptor function and improve synaptic plasticity, and we should therefore expect some kind of cognitive recovery. So this is different from the disease modification indicated by the biomarkers. We used a widely used computer-based battery of memory tests called CANTAB. These are sensitive to subtle changes in cognition. The patients had a practice test at screening, and their baseline assessment was made on day 1 before they took their first dose, and then again on day 28 after they came back after a month of dosing. There were 2 tests, a test of episodic memory, which is a type of memory that is impaired early in Alzheimer's disease, and spatial working memory. In both these tests, the patients advanced through progressively more difficult levels, and the outcome measure is the total errors across these levels. If the patient did not reach a more difficult level, the errors were imputed for that level not reached to arrive at a total error score. And because we're measuring errors, a lower score is better, and a reduction in that score indicates improvement. On slide 28 shows you the results of the test of episodic memory. So on average, the placebo patients improved by 1.5 errors. So between the baseline test at day 1 and day 28, they improved by 1.5 errors. But in contrast, the 50 milligram dose group improved 5.7 errors on average, resulting in a 37% effect size compared to that change in placebo. And the patients who took 100 milligrams improved by 4.5 errors, which is a 23% effect size. Now, we're measuring effect size here instead of significance because these group sizes are so small that we can't expect a statistically significant measure, and effect size is the way we do it here. On slide 29, this shows you the effect on spatial working memory, the other tests that we gave them. There was an impressive 46% effect size with the 100 milligram dose group and a 17% effect size with the lower 50 milligram dose group. So together, any one of these tests would indicate it's moving in the right direction, but because we have directional improvement in both dose groups on 2 different tests, it gives us a lot more confidence. It's not just a 2 plus 2, it's more like 2 plus 2 equals 10 rather than 4. The other thing on slide 30 that gives us confidence in the early cognition data is that the improvement correlated most with the reduction in p-tau 181. This is the R2 value of 0.5, and a handful of other biomarkers were also correlated. Slide 31, just to summarize, we had 98% of patients treated with Sumifilam who responded across biomarkers of AD pathology, neuroinflammation, and neurodegeneration. There were no safety issues. Sumifilam appeared to stabilize or improve memory in this small study with encouraging effect sizes and a correlation with the biomarker indicating Alzheimer's disease p-tau 181. Now, a more direct measure of target engagement and mechanism of action was demonstrated in our phase 2A study and in preclinical studies and also here in this study, but we don't have time to take you through that data today. But together, these biomarker improvements and the encouraging effects on cognition validate filamin A as a promising new therapeutic target for Alzheimer's disease.
Thank you. This is Remy Barbier. We're back on slide 33.
32. Or 33.
33 for a quick update on conclusions. The conclusions are really self-evident, I believe, if you've looked at the data. A well-controlled phase 2B study of Sumifilam showed promising treatment effects. Furthermore, phase 2B results replicate phase 2A clinical results, and the clinical results are entirely consistent with over 10 years of preclinical data, mechanism of action studies, and basic research. All in, one of the reasons why we're so excited about this drug is that overall the data highlights Sumifilam's potential for the disease-modifying effects in Alzheimer's patients. Obviously, all of these studies will need to be replicated in a large phase 3 efficacy program. Which brings us to next steps on slide 34. These studies, the phase 2B results will be published in the peer-reviewed journal. We can't tell you which ones yet. We're still having discussions, but I promise you it will be published. We've had very, very good receptions from top-notch journals. We expect to have an end of phase 2 meeting with the FDA, probably not this year because from the FDA's calendar it's getting late in the year. In addition, we also expect to complete patient enrollment in an ongoing open-label study of Sumifilam. We are starting to have the very detailed discussion around developing a study plan for phase 3, and we need to start manufacturing phase 3 clinical drug supplies. I'm on slide 35 now. A lot of intellectual protection around Sumifilam and the associated technologies. We have 7 issued patents. And now I turn it over to Eric, our CFO, for a quick discussion around financials.
Sure. Just a couple quick facts. Shares outstanding currently are 24.9 million. We also have 1.4 million vanilla warrants that are outstanding, bringing up pro forma shares to 26.3. Cash balance at June 30th was 25.3 million. And as a backdrop, thanks to the grant funding from NIH, our entire burn expected in full year 2020 is only $5 million, and we have no debt. So very well capitalized for our present needs.
Thank you, Eric. I'm now on slide 37. We're very pleased to bring Dr. Gonzalez Roja as a guest presenter, Dr. Gonzalez Roja down in Florida, has a world of experience with Alzheimer's disease, Alzheimer's patients, and I believe she's participated in 13 or more clinical trials of various investigational agents for Alzheimer's disease. So, Dr. Gonzalez, if you're with us.
Yes.
Okay, go ahead. We're listening.
Okay. My name is Dr. Gonzalez Rojas. I am one of the investigators doing the phase 2. We completed phase 2 in January this year, and I was top enroller of that trial. And currently, we are doing the open label. At the same time, the main trials that we do in our office is mainly Alzheimer's disease, which I'm very familiar with all the products out there and the mechanism of action. So usually when in the market out there is two main pathologies, mechanisms of action that companies are targeting, such as amyloid pathway and the tau pathway. And this specific trial that we're doing, I am glad to see that the mechanism of action of this medication, it will target both, because it will target the protein that would allow those things to happen. So as far as my own experience on my clinic, practical cases, on the initial trial, the 28-day trial, I didn't see much difference because of the short period of time. On the open label that we started this year also, after the second month, some patients are having some improvement. And I'm saying this, I wanted to mention at the beginning, I have no conflict of interest. This is purely my opinion of the clinical aspect of this trial and what I see in the everyday life of the practical life of the patients and their partners. So mainly after the initial months, we don't see much difference, but after month 2 and month 3, several of the patients that we have on the open labels, the partners, the companion of the patient, either the husband, the wife, or the son, the daughter, they're telling me that some patients are improving. For example, one of the patients that didn't remember much the names in the past, important person or phone numbers, and now is showing better recalling these activities. I have another patient on Cassava also that told me that they used to, they didn't know that when the patient was get confused if he took a shower or not, and sometimes used to have like a wet underwear in the shower to make sure that she didn't take a shower again that day. And the wife is telling me that this is not happening anymore, that he kind of is able to tell that if a patient took a shower or not. So these are minor samples that, we were pleasantly surprised about this. So the other thing about this trial, that the patients wanted to participate in this trial compared to the other ones, the anti-amyloid trial that I have right now requires infusion and injections. And this infusion can take 1 hour, and a lot of patients refuse to have the infusion, and this time we're having pills. What they didn't like about the pill, though, is that it was twice a day. So ideally in the future, if we want to work in a phase 3 or make it to the market, I don't know if it's possible, but ideally the patients would love to have the medication once a day rather than twice a day. So that was pretty much my impression so far. If you have any questions, I again, this is completely anecdotal. If you really want to see, you have to look at the actual numbers and what the data show. But that's my personal experience.
Thank you, Dr. Gonzalez Roja. We now would like to open it up to Q&A.
Thank you. If you would like to register for a question, please press the 1 followed by the 4 on your telephone. You will hear a 3-tone prompt to acknowledge your request. If your question has been answered and you would like to withdraw your registration, please press the 1 followed by the 3. Once again, to register, please press the 1 followed by the 4. Our first question is from the line of Jason McCarthy with the Maxim Group. Please go ahead.
Hey, Remy and team, congratulations. I know it was a long time coming, what you guys had to go through. Just a couple of questions around the dosing. The biomarker data for both the 50 and the 100 milligram group was, in my opinion, is more or less relatively similar. It trended towards, more significantly towards the 100 milligrams. But on the cognition side, on episodic memory, it seemed to favor 50 milligrams. On spatial working memory, it seems to favor 100 milligrams. How do you think about dosing going forward? Would you try to go lower on dosing, or would you even try to go higher? Because you did say that there were no adverse events related to drug. And my second question is, as you start to think about a phase 3 program, do you wait for the 100 milligram open label extension study to get you more cognition data before you make any conclusions on what that trial could look like from a protocol perspective?
Hi, yes, this is Dr. Burns. Thank you for those questions. We did notice that there was a very flat dose, almost flat dose response curve between the 50 and the 100 milligrams across the biomarkers. And this tells us that we're very nearly saturated with that 100 milligram dose group at our femtomolar target. Sumifilam binds altered filament A with the femtomolar affinity, which again is unusual, just like the femtomolar binding of A-beta 42 for the nicotinic receptor. So we believe we are at a maximum beneficial dose at 100 milligrams. We would like to stick with the 50 in going forward, both doses, but we don't see any reason to increase that dose just because we have a very clean safety profile.
I see. And in terms of the potential phase 3 design, do you wait for the 100 milligram open label data to make any permanent or final decisions on what that could look like, Remy?
No, we're not going to wait for that. We're going to design the study earlier, but the phase 3 study will have 3 arms. It's going to have a placebo, it's going to have the 50, and it's going to have the 100 to be sure that's the right direction we're going.
Okay, and do you have a strong balance sheet now. Do you initiate the studies on your own, or do you go out and potentially get a partner to go into the phase 3s?
Well, Jason, you hit the $1 million question, or I should say the $64 million question. Obviously, these are very expensive studies. We do not have the balance sheet right now to conduct these studies. Having said that, I don't believe with the strength and the magnitude and consistency of our data, I don't believe raising the money will be an issue for us. And I'll leave it at that. Obviously, as a public company, we cannot and will not provide any guidance around possible collaborations.
Of course. No, I wouldn't expect you to. I just, for my own knowledge. But congratulations all on the data. It's very exciting for the company.
Thank you.
Thank you.
As a reminder, if you would like to register for a question, you may press the 1 followed by the 4. And our next question is from the line of Marcus Hennessy, private investor. Please go ahead.
Yeah, hi there. Thank you so much for your excellent presentation. Really enjoyed it. Yeah, I am an investor in your company. I've been following the progress of Cassava for about a year now. Really exciting in terms of what you're doing with Alzheimer's and this particular drug. I have just 2 quick questions, basically. The first one is, did the company, the assay company, the lab that you used in May, early part of this year, did they acknowledge any errors or mistakes or any misinterpretation of data? Did they publicly acknowledge this? And my second question would be, I understand the phase 3 study will be rather substantial and large and protracted. When do you anticipate possibly concluding that phase 3 study and submitting your final application to the FDA?
This is Remy Barbier. Well, very forward-looking and relevant question. We'll take it in order, 1, 2. Regarding your first question, clearly mistakes were made on their behalf. And, like a lot of parties these days, it's hard for anyone to fess up exactly what happened and when and who did what. It's almost like we did our inquiry, we did our forensic inquiry. We clearly see mistakes. Others who've seen the data say it's not possible to generate this data. And so we really rely exclusively on the second bioanalysis from this study to give us comfort that our data is directional and consistent with over 10 years of research. Your second question has to do with timing. I mean, right now, I cannot look you in the eye and say our phase 3 study will start exactly on, this month of this year. So I would be even less capable to say when it ends. But typically, if you look at the record, these phase 3 studies are large, complex, expensive, and they take a minimum of 2 years.
2 years.
Minimum.
Okay, minimum of 2 years. Right. And so regarding the first question, there was no public acknowledgement of errors or misinterpretation. Basically, there was just a disagreement, essentially, between you and the analyzing lab.
Correction, there is no disagreement. It's just the data is the data. The data doesn't agree or disagree. Data it's face value. It's like looking at a coin. If the coin says 25 cents, that's the data. That people don't disagree around that. Okay. But again, in the spirit of being gracious, we're not going to name names. We have good, strong directional data in a very, very large market opportunity in a number of patients who really, really need help. For us, we're happy around the data.
Great. Well, thank you so much. Appreciate your answers.
Our next question is from the line of David Liu with Hedgehog Capital. Please go ahead. Mr. Liu, your line is open. You may go ahead with your question. Your line may be muted, sir.
Oh, sorry. Okay, sorry. Congratulations on the results. My question was just answered right before this, so thank you very much.
Mr. Schoen, it appears we have no further questions at this time. I'll return the call back to you, sir.
Okay. Well, once again, thank you. We're super excited with these results, mostly for patients. I still think it's a very exciting day for patients, and we look forward to future collaborations around Sumifilam. Thank you.
That does conclude the conference call for today. We thank you all for your participation and kindly ask that you please disconnect your lines. Have a great day, everyone.